Okay, so today we're going to talk about hair analysis. Sure, many of you are aware that hair is probably something that is used as a valuable source of evidence in a lot of crimes. This is primarily because hair is something that is oftentimes left at a crime scene. It is easy for hairs to fall off, and because they are actually composed up of cells which contain DNA, that allows for DNA analysis as well. So a criminal can unknowingly leave hair behind at a crime scene. Structure of hair can let forensic scientists determine if the hair is human or non-human, just by the way that the hair kind of looks like underneath a microscope. And as I mentioned just a second ago, hair can be analyzed using a variety of methods, including that of DNA analysis, which could be a very valuable tool. Now, what are some characteristics of hair that make it a good piece of evidence? One, it will adhere to many surfaces. Two, it easily transfers to a victim of or to a crime scene. It is very easy that if you just have sort of a brush up or even very slight direct contact with an individual, you can actually transfer hair from one surface to the next. It does have a tough outer coating, which means it's oftentimes protected by normal elements surrounding it. And physical characteristics do provide class evidence. Um, Hair, because it is composed up of human cells, does have a DNA source. Hairs with an attached follicle or root can contain nuclear DNA, and this can actually provide individual evidence in a crime scene. Without a root or follicle, it will contain mitochondrial DNA. Now, although mitochondrial DNA will not necessarily point to one particular individual over another, it does allow to reveal family relationships, and it could still be a valuable piece of information. There are various type of chemical tests that can be performed on hair, gas chromatography, and isotope ratio analysis. The gas chromatography and the isotope ratio analysis, these are two types of chemical tests in which you can sort of determine the compound composition of a sample, and allows you to really separate out all the different compounds that sort of make up a particular sample. The limitations of these tests is that it cannot determine when a hair was left at a crime scene. It can't put a timeline to it. And it also might be a secondary transfer. So the history of hair analysis goes back to the 1800s when scientists recognized the importance of hair analysis as trace evidence, meaning that they were able to sort of identify these little types of hairs that were left behind at a crime scene. With the onset of the microscope, this allowed for a more comprehensive study of hair, which was published in France in the 1900s. And we could fast forward to the 2000s, where the Innocence Project, which is focused on really kind of doing DNA analysis. This allows for changes to hair analysis findings, no conviction solely on microscopic hair analysis, meaning you really shouldn't convict somebody if you're just kind of matching hair up based on how it looks underneath the microscope. And really getting to that DNA analysis to really sort of see, is this a person that could have participated in a particular crime? This is really big for a lot of rape cases, in which case they found hair at the on the victim, and with the Innocence Project, what they're trying to do is test the DNA of the hair found, test the DNA of the suspect, and see if it's actually a match. Um, so if we kind of look at why hair even exists, kind of take a step back here a little bit. The functions of hair is that it regulates body temperature, it does decrease friction, and it protects the skin against sunlight. Human hair you have body hair, which is mostly reduced compared to other mammals. And then you have various life cycles of hair. We have what's referred to as the anagen stage, the catagen stage, and the telogen stage. And we'll talk a little bit more about these stages in 1 second. With the anagen stage, this is actual growth of the hair itself. So it's actually going through a growth cycle. The catagen stage, this is going to be a transition between growth and rest. And the telogen stage, this is when the hair is at rest. Um, okay. So if we look at the overall structure of the hair, human hair, you have a follicle. This is going to consist of the papilla. This is the area that contains the capillaries. Capillaries are the blood supply. It also contains DNA as well as those blood vessels. The hair shaft, this is the non-growing part of the hair, and this will contain mitochondrial DNA. There are different types of hair, which can vary in shape, length, diameter, texture, and obviously color. Hair from different parts of the body. We have head, eyebrows, eyelashes, beard, and mustache, underarm, body, and pubic hairs. All of which can sort of be distinguished by looking at them underneath the microscope. You can also tell if the hair was treated. Was it bleached? Was it dyed? And determining the PMI. The PMI is the post-mortem interval. This will allow to determine decomposition of a particular body if necessary. You can also look at ethnic or ancestral differences. There are key physical characteristics that are different. European, Asian, African are some examples of where you can see some of these differences. Uh, Distinguishing between non-human and human hair, there are differences in non-human and human hair. One, the pigmentations. Human hair is usually one color along the length. That might not be the case in other types of non-human hairs. The medullary index, this is the ratio of the diameter of the medulla to the diameter of the entire hair. And each type of hair in this, including human hair, will have its own medullary index. And then the cuticle, human scales are flattened and narrow compared to other types of hairs. Now, collecting hair evidence in an investigation, hair is trace evidence. As with all trace evidence, it does not necessarily provide for guilt, as it could have been left there days, weeks, years prior. Now, hair firmly does attach to clothing. I'm sure we've all kind of looked down on a shirt or something and found some hairs. Uh, Hair can leave, uh, as hair is firmly attached to clothing. This will allow for hairs to remain intact. Diagram and note exact location of where the hair was found. Photograph the hair. Package the clothing if dry with the attached hair as well. Hair that is not firmly attached can be photographed in situ. You can use clean forceps to pick up the hair. You want to place the hair in a small box or glass vial. And once in the lab, the hair can be removed. You don't necessarily want to put the hair into a plastic bag, although that's oftentimes what we see, as there's going to be some moisture in the hair, and if it's in a plastic bag that is not breathable, that can lead to actually mold, which could damage your specimen. Now, looking at the macroscopic analysis is this looking at the hair with the naked eye, and it's going to be very difficult to sort of really pick up some key characteristics, maybe beyond color with this. With microscopic analysis, you can use various types of microscopes to do various types of analyses on the hair found. Low-powered compound microscope. This is your typical light microscope that you may have used, say, in high school or in schools. Then you have your phase contrast and fluorescent microscopy, where you can look for their various structures, and the most detailed type of microscopy that you will get with a hair follicle is going to be the electron microscope. As we said earlier, you can do chemical analysis on the hair. Anything that's ingested or absorbed through the skin could have potential traces or chemicals in the hair. Hair does not readily decompose, and it can also establish a timeline based on where you might find the chemical along the growth of the hair. So going back, for example, drug analysis, uh, testing people to see if they're taking certain types of drugs or on certain types of drugs. They can take some hairs and do drug tests on that, essentially. Um, as far as establishing a timeline, depending on where they find the chemical, is this towards the end of the tip of the hair or is this closer to the root? That can sort of give a timeline on maybe when a particular type of chemical was used or ingested, for example. And because hair does not readily decompose, you can also sort of go back and potentially exhume the body, and you might find some hair follicles there, which can be further analyzed for chemical analysis. The nice thing about the hair follicles is that it is really a snapshot of that chemical when uh, the hair follicle was actively growing, and if there was chemicals in the bloodstream, those very well could show up on this hair analysis. DNA analysis, hair that contains the follicle, the root, that you can perform DNA analysis on, and this will give you individual evidence. There have been several advances in hair analysis as well. For the neutron activation analysis, this actually um, is an analytical technique that relies on the measurement of gamma rays emitted from a sample that was irradiated with neutrons. The rate at which the gamma rays are emitted from an element in a sample is directly proportional to the concentration of that element. So again, this really just helps to sort of determine the composition of that sample. With isotope ratio mass spectroscopy and liquid chromatography, this enables high precision measurement of stable isotope content in the sample. Typically, the measurements will target hydrogen, carbon, nitrogen, and oxygen analyses. Although elements with masses up to and including sulfur can actually be measured using this technique. And then another type of hair advances in hair analysis is actually, in addition to DNA analysis, we can do protein analysis. Protein analysis can be just as specific as DNA analysis, depending on the samples. So in summary, hair can be a common and often overlooked form of evidence. It is characteristic shared by by all mammals. Microscopic hair analysis is considered to be more subjective form of analysis, and hair consists of a follicle, which is a root embedded in the skin that produces the shaft. The hair shaft is composed of the protein keratin. which is a very tough protein, and it allows for waterproofing of the sample. Hair varies depending on where on the body it originates. The development is divided into 3 stages: anagen, catagen, and telogen. Anagen, again, is active growth. Catagen is transition. And telogen is when the hair follicle is at rest. Hair from different parts of the body look very different from each other as well. Human hair is different than other mammalian types of hair. It must be properly collected and stored. You can't, as I mentioned, you cannot put hair follicles inside of a plastic bag. Hair is chemically analyzed using the various types of techniques we talked about on the prior slide. And these new advances can provide clues on where one particular individual actually lives. The hair shaft can provide mitochondrial DNA for class evidence. The hair follicle provides nuclear DNA for individual evidence. And microscopic hair analysis evidence is still used, but it is considered to be a more subjective form of evidence versus that of DNA analysis. Okay, and that is it for this chapter.